Good morning, all of you, first of all. Done. See. Okay, not audible. Is my voice audible? Check it there. Okay, good. Everything fine. See? Yes. Okay. Now. Let me begin the NEET perspective. See? I would like to say only one thing. Don't analyze why it happened, how it happened, and what is happening to us. Don't analyze anything. Just keep that thing side by and focus on how to respond to it. If you're thinking, I have done very well in the past exam, what would be happening in the future exam? What would be my future? Remember, things will not happen by luck. They always happen by work. So, forget about what happened. Let us restart. Keep the mind fresh. Delete everything, whatever happened, and don't watch these news unnecessarily. How it happened, how to do this, that. Don't watch the news. Don't waste your time. Just focus on 1 hour other day. They have to conduct NEET exam. That is confirmed. Whatever the difficulty that we have faced, that's okay. But it may be after 1 month, after 20 days, even after 2 months, they have to conduct exam. For that, you have to ready and don't waste your time. Think that whatever the NEET exam that you have written on May 3rd, it was one of our face exam. Regularly we write, but we have done some kind of work for that, traveling here and there. Like that, think about it. Delete that exam and let us practice for the coming exam as we started these face exams enthusiastically. That means before starting of the face exams, whatever the mindset we had, let us start with the same mindset. Daily you have to do one thing. Allot some time to biology. I am saying at least 4 hours for biology. 4 hours. In that 4 hours, 1 hour for practicing bits for a particular lesson. The another hour you have to spend it for practicing NCERT book. I think you had experienced on the May 3rd. There is no any question, especially in biology, which hampered your flow. It was moving fluently and very smoothly. The same experience we will expect in the future also. So, read the NCERT book. Again, one more idea the people are facing now. I was getting around 350 marks in the past NEET exam which was conducted on May 3rd. Should I get the same number? Definitely you will get the same number. The only thing is you have to revise it. If you left the biology like that, okay, I have done very well in biology. In the past exam, I couldn't able to cope up with chemistry and physics. So I will focus more on there because already I am able to score 350 in biology. So I would be able to focus more on physics and chemistry. Don't get this idea. Stay with biology. I'm not saying stay with only biology. That's another bad thing. Stay with biology. Allot some time in your day. Make a schedule. In that schedule, biology must occupy at least 5 hours of your study. 5 hours. I'm insisting that. 2.5 hour botany, divide according to your schedule. 2.5 hour botany, 2.5 hour zoology, we make it. In that, practice only NCERT book, only NCERT, including summary, the introduction, and also the scientist who associated with the particular lesson. Everything practice. And also, after practicing, don't leave that, which means I have done practice the NCERT book, it's fine. No, that's not fine. At least for every lesson you open some bits, but don't practice the same variety of bits. You open some bits. If they give DAT exam, definitely practice the DAT exam. And if they give any kind of papers to you, if they forward or if you come here, they physically provide the paper to you, all those papers, make sure that you are practicing well. Don't take rest. This is the time to again rejuvenate the fallen edge of our practice. Of course, you might be relaxed for a while. That relaxation is over. Again, as a soldier, when the enemy attack, even they are in the sleep, they have to get up, and then without even washing their face, they have to take the gun. This is our schedule now. If you are not ready like that, you have to have some kind of, difficulty to be faced at the time of next NEET. Let us have some glance. Remember, in regular days, I will do regularly some kind of revision. One particular lesson we will take, and whatever the things frequently students started to forget, or else those areas where students are facing little bit difficulty in order to cope up the particular sentence or the particular idea which was exposed in the NCERT book, but the student could not be able to exactly catch that idea perfectly. That thing I will like to explain. Okay, after my explanation, you feel comfortable with the NEET exam, you will feel comfortable with the NCERT book, then automatically you would be able to do it somewhat faster. Remember, if you are doing NCERT book by yourself, it will take some time. But listen one time class, I will go only NCERT things, because right now, if we are having some lot of things that may not going to work out, so I will go line by line regarding to NCERT book. Wherever there is a necessity, I will give the relevant explanation. You try to follow that explanation. After that, by your own way, try to read NCERT book line by line, and what they want to say in that particular area, try to understand and try to cope up with that. Then after that, practice bits. It's not difficult for you, because you have already done many difficult things. So let us start the story. Okay, now I am going to begin anatomy of flowering plants. Over there, here, what is anatomy? The anatomy is study of internal organization of an organism. Internal structure we are going to study. We have animal anatomy and plant anatomy. Animal anatomy is very difficult, because there are variety of tissues, variety of organizations, etc. would be there. But plant anatomy is very simple. Plants are simple organization bodies. But in the anatomy, you might have several things like cell biology and also cytology, etc. But we don't touch all of them. Just we will talk about internal organization of a particular plant. Especially, whatever the anatomy they try to explain in this lesson, they try to focus only on angiosperms. They don't want to talk about the lower kind of plants. Here and there they have given some words that are associated with gymnosperms. For example, these are not found in gymnosperm, they are found in only angiosperms, but the main focus is on angiosperm anatomy. Okay, then let us check. They are saying, what is a tissue? Tissue is a group of cells. This is what we generally believe, but tissue is a group of cells which are having common origin and usually performing common function. They must be originated from same mother cell. For example, if it is a cell, it would be divided and it is going to produce new cells. Okay, new cells. Then the mother cell, which is constantly dividing and forming various types of cells, several varieties of cells, and all together, all these cells, if they are working together, we will call them as tissue. Be careful. Those daughter cells which are coming out of a parental cell need not to be identical. I mean, if a tissue consists of 3 to 4 types of cells, if I say them as tissue, all those cells must be having common origin and they collectively conducting common function, but they need not to have common structure. Keep it in mind. Okay, if I say xylem, xylem has 4 types of cells: tracheids, vessels, fibers, and parenchyma. But if I say all of them collectively called one type of tissue, they all must be originated from a common mother cell, and all of them collectively should be conducting a common function, but they need not to be having common structure. Keep it in mind. A tissue cells should not be having common structure. They must be having common origin and common function. Next one. They may be classified mainly meristematic, who are able to be divided, and permanent. And the permanent cells could not be able to divide, and meristematic cells could be able to divide. Let us check meristematic cells. Who are the meristematic cells? The cells which are able to be divided are called meristematic cell. Remember that meristos means to be divided. Remember, I am going to take the classification of meristematic cells. Meristematic cells over there. Meristematic cells, first of all, based on origin, from where they are coming. If they are coming from embryonic meristem or embryonic cells, embryo, or they will also say sometimes embryo means procambium. Okay, then the embryonic origin meristems are primary meristem. Their origin is primary. From the beginning of the body onwards, they have been there in our in the plant body. That's why they are called primary. Then after the primary, if you talk about what are the types of meristems in the plant body are primary. They are apical meristem, and also there are meristems in the name of intercalary meristem. Observe this. Intercalary meristem is also primary meristem. Keep it in mind. If there is a meristem that is universal for all kinds of plants, whether you take an angiosperm, gymnosperm, whoever it may be, if there is a tissue that is universal, whoever conducting any kind of different growth like secondary growth and who are not conducting secondary growth, whatever it may be, if you come across a tissue that would be universal, that universal meristematic tissue must be apical. The apical meristem would be shoot apical meristem and also root apical meristem. Okay, intercalary meristem. This meristem is not universal. The intercalary meristem would be typically seen in case of grasses. In the grasses, what happens? It is present in intercalary. Intercalary means, callow means stem. Within the stem, in between the nodes, a special location, this intercalary meristem is present. If there is any herbivore grazed the grasses, the broken part of the grass would be regenerated with the help of this intercalary meristem. Apart from that, intercalary meristem does not have any other kind of work. Next one. When we talk about origin, there is another origin is there, secondary meristem. Secondary meristems. Remember, the secondary meristems are those meristems which would be found in the case of only those plants which are performing secondary growth. If a plant has secondary growth ability, like in case of gymnosperms, as the dicot angiosperms, they can able to conduct secondary growth. Those plants which are able to be conduct secondary growth will have secondary meristem. What happens in the secondary meristematic category? We will have, okay, interfascicular cambium or interfascicular cambium, and also this one commonly call it as vascular cambium, and also cock cambium. Okay, this cock cambium is also called phellogen. We'll talk about it. So secondary meristems, they are developed by a process called dedifferentiation. They are developed by a process called redifferentiation. That's about origin. Then go to another kind of category, location, where they are present. Location would be, again you will have apical, intercalary, and also lateral. Observe this: apical, intercalary, and lateral. These are locations on the plant body. Over there, apical meristem, you know already, root apical meristem, shoot apical meristem, and intercalary meristem, if you are talking, it has only one name. I am giving another name, that is vascular cambium. Okay, and lateral meristems. Who are lateral meristems? You will have here, sorry ma, we will have the story here, like, okay, intercalary meristem, we will have this one. That one is first lateral meristem, cork cambium, and also lateral meristem you would be having interfascicular cambium. Okay, and these meristems would be present in lateral sides of the plant, especially if there is a root or stem. Lateral sides they are present. That lateral side presence will make them to make the plant to be enlarged laterally. Is it okay? So in this, based on the location, based on the origin, if you compare the meristems, you will have some situations like, when we talk about all of them over there, for example, the only primary meristem which is present in lateral location, that is called, okay, intrafascicular cambium. Sometimes it is also called vascular cambium. This is about based on their location and based on their origin. Simply remember, okay, when there is a meristem that is present in apex, apical meristem, either it is shoot apex or root apex. Next, when you go to lateral meristem, they would be present on the lateral side of the plant body. The lateral side, they would be helping for enlargement of the stem as well as in the root in case of the dicot and gymnosperms. Okay, then come back to this diagram. Observe, this is, they wanted to show root apical meristem and shoot apical meristem. Observe, this is the area where shoot apical meristem exactly present. Okay, this shoot apical meristem, if you talk about its growth, gradually it will grow like that. Some amount of shoot apical meristem would be left over at the axil of the leaf. Imagine this is leaf. If they are talking about a newly formed leaf, that newly formed leaf we call it as primordial leaf primordium, or the leaf that it has just now originated. It has not yet started any kind of extensive growth, or it is not showing any typical properties of a leaf, but just now it has started. That's called leaf primordium. Some amount of shoot apical meristem would be left over at the axil of the leaf. This is called axillary bud. If somebody is asking, axillary bud is primary meristem or secondary meristem? It is primary. Then what is the use of the meristem would be left over at the axil of the leaf? This axillary bud, later it could be able to produce a branch. It can produce a branch, or if it necessary, it even could be able to convert in the form of flower. That's about axillary bud. Remember, axillary bud is nothing but a leftover shoot apical meristem. In that case, could be able to find any axillary buds on the root apical meristem, and lateral meristems or intercalary meristem. None of them could be able to produce axillary bud. Axillary bud could be coming from only one type of meristem, that is called shoot apical meristem. Got it? Then here they are talking about root apical meristem. What do you mean by differentiating vascular tissues? Actually, if you talk about vascular tissues, xylem and the phloem, xylem and phloem have been originated from the shoot apical meristem in the initial growth of the shoot. At the time, if you talk about the shoot apical meristem, slowly some amount of shoot apical meristem started to differentiate and convert in the form of xylem and phloem. That xylem and phloem together is called differentiating vascular tissues. Then come back to the root. Root apical meristem, some amount of root apical meristem growth. Observe this. Some amount of root apical meristem would be go away from the root tip and it would be differentiate in the form of root cap. Be careful. Always the root cap would be present away to or towards the proximal and distal directions. If I move towards the center of the plant, that's called proximal. I am moving away from the center, that should be a distal one. The root apical meristem is the one which is going to be considered here, providing root cap. The root cap would be present distal. Next one. Some amount will be converted in the form of initiation of central cylinder. What is central cylinder? In the center we will have xylem and phloem and other kind of supporting tissues. They will be coming from root apical meristem. And protoderm. Protoderm means new epidermis. New epidermis is called protoderm. And also cortex, central cylinder, all are coming from root apical meristem. This is about the diagram. Then go back to, look at this. Here I have seen many students are getting confused. The meristem which occurs between the mature tissues is known as intercalary meristem. That's okay. If you are talking the intercalary meristem, is it a primary or secondary? It is primary. Keep it in mind. Okay. It occurs in grasses and other kind of, okay, you can understand that. But you should remember, intercalary meristem is a primary type of meristem. It's not secondary meristem. Next one. Secondary lateral meristem. All those things I have explained to you. Next one, permanent tissues. What do you mean by permanent tissue? The tissue that could not be able to divide. That's what primarily we understand. That's called permanent tissue. But one thing you have to remember, the primary tissues are functional. They are capable to do the work. Meristematic cells do not have any work at all. Keep the statement perfectly understandable like this. If you talk about any meristematic cell, the meristematic cells could be able to divide. But the dividing cells are not able to conduct any kind of typical work or typical physiological function that is essential for the survival of a plant. Which means meristematic cells do not conduct photosynthesis, they can't store the food, they can't able to transport the food and water to various organs. They don't have any work. But permanent cells are functional, permanent tissues are functional. The making of the process of permanent tissues is called differentiation. Observe the story. Differentiation. What do you mean by differentiation? If you have primary meristem, the primary meristem would undergo some changes. The changes will make the primary meristem to be converted in the form of permanent tissue. So the permanent tissues could be able to help the plant to survive. Got it? Then come back to the story. Who are the permanent tissues? Remember, the permanent tissues, here we are talking about primary permanent tissues. Whatever the list of tissues that they have explained here, they are primary permanent tissues. We do have secondary permanent tissues in the name of cork, etc., etc. They would be discussed in later. But here, remember, primary permanent tissues, if you talk about them, simple permanent tissues, and the next one, complex permanent tissues. What's the difference between the simple and complex? The simple tissue would be having only one type of cell. One type of cell. What did I say about a tissue? Whether they are similar, dissimilar, they must be originated from common mother cell, and collectively they have to conduct a common function. Such kind of group of cells we call it as tissues. That's what the definition of tissue says. But when it comes to simple permanent tissue and complex permanent tissue, simple permanent tissue would be having all the cells are similar. They don't have variations. Complex permanent tissues are the one who are having differentiations. That means a group of cells, even though they have differences in their structure, but collectively they are going to conduct a common function. That phenomena we call it as complex permanent tissues. Simple permanent tissue, three we have. One is Parenchyma, Collenchyma, Sclerenchyma. Next, complex permanent tissues we have Xylem and Phloem. Then if you talk about simple permanent tissue, the first one, okay, Parenchyma. Remember, the Parenchyma is also called most fundamental tissue. Fundamental tissue. Why did you call it as fundamental tissue? You can see a plant without Collenchyma. We can see a plant without Sclerenchyma, and even some cases we can see a plant body without Xylem and Phloem also, because from pteridophytes onwards we have seen development of Xylem and Phloem. But we couldn't see a plant without any Parenchyma. That's why it is called as fundamental tissue of the plant body. It conducts various types of functions. First of all, let us see its physiology. If you talk about here, this is the Parenchyma. The shape would be vary. You cannot have a conformed one particular variety of shape regarding to parenchymatous cells. Their shape is going to be vary. And at the same time, if you talk about from external to internal, they have intercellular spaces. Intercellular spaces. These intercellular spaces sometimes very enlarged, and even they may be filled with air gaps. If the intercellular spaces filled with air gaps, they are called as Parenchyma. Parenchyma. Okay, this Parenchyma will be helping for buoyancy, which means they can able to float on the surface of water, or if the plant wanted to maintain its buoyancy, that buoyancy could be maintained with the help of Parenchyma. Remember, if a plant organ wanted to store air, that would be stored only in the intercellular spaces. No plant would store air within the cell. Only outside the cell, air could be stored. Yes, a plant body could be able to store some sort of air if it is necessary. But the plant organ never ever able to store air within the cell. The plant organ will store air only in between the cell in the name of interstitial spaces, or in common words we can call it as intercellular spaces. Next one, come inside. If you talk about their cell wall, they have cell wall of course, but the cell wall is primary. What do you mean by primary? If you are talking about primary cell walls, the primary cell walls are generally made up of, okay, cellulose, hemicellulose, primary cell wall, and they may have vacuoles and various other kind of things, and they do have chloroplasts if they wanted to perform photosynthesis, and the remaining story well known to you. Let me read one more time. Parenchyma is the major component of within the organ, very important. That's why it is called fundamental tissue. The cells of the parenchyma are generally isodiametric. Isodiametric means they have various kinds of shapes. They may be spherical, oval, ground, polygonal, or elongated in the shape. Their walls are thin and made up of cellulose, but they do have some amount of hemicellulose. General chemical is cellulose. One blind thing remember, if there is a cell wall which is made up of primary cell wall construction, the primary cell walls are made up of cellulose, nothing else. This is blind thing, you remember. Next one. They are either closely packed and have some intercellular spaces. Parenchyma perform various functions. If they perform photosynthesis, that's called as Sclerenchyma. Sclerenchyma. Example, okay, mesophyll cells. Storage parenchyma. Various kinds of organs in root, stem, various places, food will be stored. And also secretion. Secretory parenchyma, you know, some plants would secrete scents, some plants would be able to secrete various kinds of gums, resins, etc., etc. They would be stored in parenchyma only. One more thing, somebody is storing something, okay, that would be none other than our parenchyma. No other kind of tissue is having the ability to store any kind of food. Is it okay? Next one, Collenchyma. This is exclusive. This is not universal. This is exclusive. Mainly found in the most of the dicotyledonous plants below the epidermis. Below the epidermis means hypodermis. It's not universal. Keep it in mind. Hypodermis. And why does it present in hypodermis? It is going to give some sort of mechanical stability to the young stems. Gradually, when the stem grows, its requirement of Collenchyma would be reduced, because other than Collenchyma, some more variety of tissues would be developed in the plant in order to provide mechanical stability. So, if you have stronger bodies, you need not to have Collenchyma. That's why Collenchyma would be gradually disappeared in older bodies, but it will be present in younger stems, and also it is present in the petioles of the dicotyledonous leaf, especially in the hypodermal area. What is the use of that petiole Collenchyma? Petiole will be fluttering in the wind. That means while the leaf is fluttering, the petiole must be providing some sort of mechanical stability. That mechanical stability would be provided by the chance of Collenchyma. Got it? Next one. When you talk about Collenchyma, you have to remember that it is ready to be provide mechanical strength. Okay. How does the Collenchyma would get mechanical strength? Remember, Collenchyma cells are without any intercellular spaces. They don't have any intercellular spaces. If the spaces are less, that area would be filled by cellulosic or other kind of cell wall material that would give strength to the plant body. Next one. In the corners of the cell wall, okay, there would be a deposition of hemicellulose, pectin, cellulose also. Okay, cellulose is a regular thing, be careful. Deposition of cellulose is common for every cell wall. Without any cell wall, without any cellulose, cell wall will not be formed. But in addition to cellulose, hemicellulose, pectin will be deposited at the corners. As a result of the deposition on the corners, that is going to make the plant organ to be more stable. This is called as, okay, a kind of mechanical strength. Collenchyma cells are oval shape is, okay, they can assimilate the food. What do you mean by they can assimilate the food? Remember, Collenchyma is nothing but a modified parenchymatous cell. When it is modified to provide mechanical stability, but even by having chloroplast in their cell cytoplasm, it can able to perform photosynthesis. That's why it can be assimilate the food. That's the reason why it is called photosynthetic mechanical tissue. Okay, intercellular spaces are absent. They provide a mechanical support to the growing organ like young stems and petioles of the leaf. Be careful. Within the plant body, if somebody is performing photosynthesis, that one should be parenchyma, so called Sclerenchyma. If other than parenchyma, if anyone perform photosynthesis, that should be Collenchyma. Collenchymatous cells are specialized kind of cells who provide mechanical strength. In addition to provide mechanical strength, they do also conduct photosynthesis. Next one, Sclerenchyma. Here remember, Sclerenchyma are dead cells. They are dead. Why are you saying they are dead? No protoplasm. They don't have any kind of protoplasm. No plasma membrane, no cytoplasm. Now here I could see lignified walls having narrow, numerous pits. What do you mean by pits? If there is a cell wall, inside the cell wall some small minute gaps are present. These gaps we call it as pits. Okay, the pits. Pits would be present whenever the cell wall has got more thickened, more thickness of the cell wall. If there would be some sort of gaps would be generated. Those gaps we call it as pits. There are variety of pits. Let me go forward and then I will elaborate them. On the basis of variation in the form, structure and origin, Sclerenchyma may be of either fibers or sclerites. Remember, both are dead. Fibers are thick-walled, elongated, pointed cells. Underline this. They are pointed cells. They have this kind of arrangement. They are pointed cells. Sclerites are spherical, oval, cylindrical, highly thickened dead cells. But when it comes to cell wall thickness, imagine I am keeping the cell wall here. When it comes to cell wall thickness, fibers they do have thick cell wall. I am not denying that. But the thickness of sclerites, cell wall will be more thick cell wall or thickened cell wall when compared to fibers. Both are dead, but the shape only varies. Sclerites are spherical and cylindrical, highly thickened dead cells with very narrow cavities. Remember, they are not saying cytoplasm anymore. When do we say cytoplasm? If a cell is live, conducting living functions, then I will have cytoplasm or protoplasm inside the cell. If the cell is dead, the dead cells do not have protoplasm. They have an empty space inside the cell. That empty space we call it as lumen. They are commonly found in which one? Sclerites are commonly found in fruit walls of the nut. Important. Pulp of the fruits like guava, pear, sapota, because they have some grainy structure. Seed coats of legumes and the leaves of the tea. Okay, Sclerenchyma provide mechanical support to the organ. Remember this. If somebody is asking, okay, somebody saying this statement to you, the sweet nature of sapota is due to Sclerenchyma. Right or wrong? The sweet taste of pulp of sapota is due to Sclerenchyma. Right or wrong? Okay. Absolutely wrong. That sweet nature is due to Parenchyma. The sweet nature is due to storage of some kind of material that's called sucrose as well as fructose. Remember this thing. The Sclerenchyma would be present in the fruit pulp of guava, sapota, pear, etc. But in them, Sclerenchyma is not the one which is responsible for their taste and storage of material. Sclerenchyma just to provide mechanical stability. In addition to Sclerenchyma, they do have Parenchyma as well. Keep it in mind. Okay. Complex tissues. Who are the complex tissue? The complex tissue should be our xylem and phloem. Tell me, if you talk about xylem made up of what are the types of cells? It is made up of tracheids, okay, vessels, fibers, and also parenchyma. Okay, in this tracheids, vessels, and fibers all are dead. Since most of the members of the xylem are dead, that's why I will call xylem as dead conducting tissue or dead tissue. Correct. Only one member in the xylem who is alive, that is parenchyma. The only member living. Whatever the typical parenchymatous cells that we have studied, the same story would be repeated with respect to xylem parenchyma. Let us talk about the differentiations. If you talk about tracheids, these are the main water conducting elements in gymnosperms. Main in gymnosperms. Why are you saying they are main in gymnosperms? Because vessels are absent in case of gymnosperms. Is it okay? The main conducting elements in gymnosperms are tracheids. Next one. If you talk about tracheids and vessels, both can able to conduct water. But when it comes to angiosperm, vessels are the main water conducting elements because they do not have tracheids. I mean, they do have tracheids, but their importance is less significant in case of angiosperms. Then what about fibers? Remember, fibers are mechanical strength. Mechanical. One thing, if they are asking, xylem conduct water. Yes, xylem conduct water, but in that xylem, the main water conducting elements are vessels in case of angiosperm, tracheids in case of gymnosperms. But all the members of the xylem do not conduct water. Keep it in mind. Vessel, tracheids, vessels, fibers, parenchyma. Out of all, if angiosperm, vessel, if gymnosperm, tracheid, but all of them are not conducting water. But why did you say xylem is a kind of tissue that conduct water? Because all of them to be present collectively, and they all originated from a common mother cell. Keep it in mind. For the xylem, tracheids, vessels, fibers, parenchyma we need. All of them have come from a common mother cell, that is the one which is making all the cells to form, but out of them some are habituated in order to conduct water. Okay, let me go one by one the details. Xylem function as a conducting tissue for water and minerals from root to the stem and leaves. Remember, direction must be specified. Xylem function as conducting tissue for water and mineral from root to stem and leaf, because xylem conduct water and minerals unidirectionally. Next one, it also provides mechanical strength to the plant body. Whatever the name you are providing in the name of wood, you know, wood. The wood is nothing but our xylem. It also composed of 4 kinds of elements, namely tracheids, vessels, xylem fibers, xylem parenchyma. Gymnosperms lack vessels in their xylem. Tracheids are, be careful that gymnosperms lack vessels in their xylem, because vessels absent, that made tracheids would be more significant in them. Okay. Tracheids are elongated or tube-like cells with thick and elongated walls and tapering ends. This is a tracheid. Observe this. This is a tracheid. Tracheids will have a long tube-like elongated cells. They have edges which are pointed, chisel-like edges like this. One tracheid member would be connected with another like this. This is the way. If they wanted to conduct water, they have some connections. The connections in the cell walls, we call it as pits. Next one, they have secondary walls. Tracheids will have not only tracheids, whoever it may be strong, all the strong cells will have secondary walls. The secondary walls will make them to have some kind of pits which are called bordered pits, and the pits are making the transport of water from one cell to another. Next one, when the tracheids are developing, the thickness of the wall due to deposition of lignin. Cell walls will be having lignin deposition. Wherever there is a lignin deposition, that cell is having secondary wall. Keep this word carefully. If you have seen a plant cell which has cellulose, hemicellulose, pectin, that plant cell would be having primary wall. If it has the primary wall would be able to provide some sort of strength, but secondary wall is the wall which could be able to provide more mechanical strength. In order to achieve the secondary wall, there will be a deposition of a chemical. That chemical is called lignin. Remember, I have a cell. This is like this. Okay. And I want to make this cell to be more mechanically strong. In order to make the cell to become more mechanically strong, what I have to do? I have to deposit lignin. The mode of lignin deposition varies from one group of plant to another. Sometimes they will be depositing this lignin inside. One more thing, always whenever we are going to deposit any material to the cell wall, depositions will not come from the external side. Depositions will come from inside, which means if it is a cell, here I want to make this cell wall to become strong. Here I will deposit. I will never deposit from the top because cell cannot able to control what is happening outside. Cell will only control what is happening inside. A cell wanted to be more strong. The more strength would come from inside deposition. Is it okay? Then when it started to deposit, sometimes the depositions will be patches. We will add some amount of lignin from inside, like a patch. We are going to add some patches here and there. We are going to attach some amount of lignin. That is patches. Sometimes it would be angular. That means inside. You remember? Inside area, I wanted to deposit the material. So one side to another side, I am going to make a ring-like arrangement of lignin. This is called angular. Sometimes it would be spherical. We should differentiate this one. Angle. I mean one ring I am going to add. That is angular. This is spherical. Like that, depositions of the lignin would be vary from type of body, one type of plant body to another type of plant body. Is it okay? Getting the idea? Like that, there will be a deposition of the lignin. That's why you can see in one vessel, the depositions are going to be patches like. In some other vessel, the depositions are going to be, reticulated. Reticulated means unevenly they are going to be deposited. How they will be deposited? After the deposition only you will know. That's called reticulated deposition. Sometimes they will be deposited in the form of ring-like manner. Okay? And sometimes they will be deposited in the patches wise. Whatever it is, the depositions are going to be varies. Because of the deposition only, the lumen of the tracheids will become narrower. If narrow the lumen, thicker the cell wall, ultimately the cell is going to become more strong and mechanically stable. You got the idea? That's the reason why we will have this kind of arrangement. But you see the vessel. Vessel is somewhat similar to tracheid. But the difference is vessels will have more lumen. That means they are pipe-like arrangement. Remember, I have one pipe and on the top of that pipe, I will have another one. Like that they will be arranged. This is the vessel. Within the vessel, there will be a plate which is called end plate. Those plates are having some perforations. Perforations means gaps. By the presence of the gap, the water could be easily flow from one vessel member to another vessel member. Be careful, vessel is not one cell. Vessel is all the cells which are working to transport water together. I will call it as vessel. Vessel is not a one tissue. Vessel is a group of cells. Okay, vessel is not one cell. Vessel is a group of cells. Over there, this vessel, I will call it as vessel member. This is also vessel member. Another vessel, 3 I have written. Each one is called vessel member. So one by one, they will be connected together with the help of perforations in their common wall. If you keep 2 dabbas, one on the top of another, their both walls will be sharing. That area, they have holes. Those holes are called perforated end plates. As a result of that, they could be able to transport the material or especially water and minerals from one area to another. Next, let us complete the xylem at least. Xylem functions as a conducting tissue for water and minerals from root to the stem and leaves. It also provides mechanical support to the plant organs. It composed of 4 different types of elements, namely tracheids, vessels, xylem fibers, xylem parenchyma. Gymnosperms lack vessels. Tracheids are elongated tube-like cells with the thick and lignified walls. Underline that. If lignin is not there, they could not be able to provide mechanical strength. Next one. And they have tapering ends. Tapering means pointed ends. These are dead without protoplasm. Okay. The inner walls of the have thickening with varying in form. The inner wall, nothing but inside the tracheids, the deposition will be happen. There is lignin deposition especially. In flowering plants, tracheids and vessels are the main water conducting elements. Why they are saying that? Because tracheids, understand this. In flowering plants, tracheids and vessels are the main conducting water transporting elements. Vessels are the main, but along with the vessels, tracheids also conduct water. Be careful. If they are saying in the plant in angiosperm, tracheids do not conduct water, that's absolutely wrong. Tracheids also conduct water, but the efficiency of the tracheids is less when compared to vessels, but tracheids do conduct water. Next one, vessel is a long cylindrical tube-like structure made up of many types of cells called vessel members. Vessel members, each with lignified vessel members. Vessel is a long cylindrical tube-like structure, many vessel members, each with a lignified wall and large central cavity. The vessels are also divided of protoplasm. Vessel members are, they are also divided of protoplasm, means they do not have protoplasm. Vessel members are interconnected through perforations in their common walls. That's what I have said there. Presence of vessel is a characteristic feature of angiosperms. Why gymnosperms do not have vessels? Xylem fibers have highly ligand cell walls, obliterated central lumen. Obliterated means disappeared, or completely destroyed. Remember, I have a cell like this. I can say there is a lumen inside. Same cell, I will deposit more amount of material over there. Is there any lumen inside? That lumen has been destroyed or obliterated due to deposition of lignin centrally. You got it? That's the reason why we will have something like fibers. Xylem fibers, these either in the septate, very important. These are septate or aseptate. What do you mean by septate or aseptate? If you take a fiber, okay, in the fiber, this area I have deposited. After the deposition, there is a gap. Again deposited. Again there is a gap. Like that, you can see there are cross walls. If cross walls are present, that's called septate. If cross walls are absent, that's called aseptate. Parenchyma are living made up of cellulose. They store food material. Why they are saying made up of cellulose? Lignin is absent in them. They store food material in the form of starch, very important. Fat and other substances like tannins. Be careful. Tannins are those kind of things which are providing woody smell. They are variety of secondary metabolites. If you remember or not, there is a name called tannins in secondary metabolite list. What is this tannin? Tannins are a variety of organic substance. They are the one who are providing woody smell to the wood. Once the wood is perfectly found, the woody smell of the wood is coming due to the presence of tannins. Okay, the tannins are very important material which are giving some sort of strength also to the, when you talk about hardwood, you might have studied the word called hardwood. In the hardwood, they say it will store variety of organic substances. Okay, the organic substances which are mainly stored by them is nothing but tannins. Be careful, tannins are not found in phloem. Tannins are found only in case of xylem. Phloem will also store food material and other substances in the parenchyma, but in the phloem parenchyma you will have resins, you will not have tannins. The radial conduction of water is also takes place with the help of parenchyma. Radial conduction of water is what? From the center to periphery. Is it okay? Somebody is asking, does a plant store air in the vacuole? Never. We will have air vacuoles, air sacs only in case of bacteria. Very good thing. Only in case of the bacteria you will have air filled sacs. That too not in all bacteria, only in case of the photosynthetic bacteria. They are called gas vacuoles. But plant never ever store any kind of thing like air. Any more questions like that? Come on. Okay. Any more? Any more? Okay, shall we continue? Come on. Next one. Xylem fibers, they are talking about fibers here, xylem development here. Primary xylem is of two types, protoxylem and metaxylem. Imagine xylem is newly forming, just like they are saying xylem is newly forming. While a xylem newly forming, first of all, this is newly formed xylem and xylem gradually get matured. A newly formed xylem from its mother cell is called protoxylem. The matured one which is ready to be conduct water is called metaxylem. Okay? Imagine, important. The protoxylem lies toward the center and the metaxylem lies in the periphery. This type of arrangement is called end arc. That is, end arc is found in case of stem. In root, they have protoxylem towards periphery, metaxylem present towards center. This is called as protoxylem periphery, metaxylem center. This is called exarch protoxylem. In that condition, this growth is from center to periphery. This growth is peripheral to center. Peripheral to center, this is centrifugal growth. This is called centripetal growth. Okay? Phloem transport the food material usually from the leaves to other part of the plant. Observe, phloem, you know, it has sieve tubes, companion cells, and also tracheoles, like that structures would be present. Let me confirm one by one. Sieve tubes are also long tube-like structure arranged longitudinal and are associated with the companion cells. Why? Because they don't have a nucleus, and the nucleus needed for the maintenance of pressure gradient, and altogether always sieve tubes will be associated with a special variety of parenchymatous cells. They are called companion cells. Okay, their cell walls are perforated in sieve-like manner. Cell walls, end walls are nothing but like this, perforated, sieve-like manner. That's why they have got the name sieve tubes. A mature sieve tube element possesses peripheral cytoplasm and a large vacuole but lack nucleus. That's why they are called enucleated. Enucleated. What do you mean by enucleated? Nucleus has been removed. The functions of the sieve tubes are controlled by the nucleus of companion cells. Companion cells are specialized parenchymatous cells which are closely associated with the sieve tube elements. They are closely associated with sieve tube elements. Okay. The sieve tube elements or companion cells are connected by pit fields. What do you mean by pit fields? If it is a companion cells, this is a sieve tube. Both are connected together by a small gap present in the cell wall. That gap present in the cell wall is called pit. The sieve tube elements companion cells are controlled by pit fields present in their common longitudinal walls. The companion cells help in maintain the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering, cylindrical cells which are dense cytoplasm and nucleus. That is phloem parenchyma story. The cell wall is composed of cellulose and it has pits through which plasmodesmatal connections. What is the pit? Pit means, be careful, if you come across a word called pit, pit means the one which is having a small gap in their cell walls. That's called pit. From one cell to another cell, if you are talking about there are cytoplasmic connections, the connections are called plasmodesmata. Plasmodesmata is a protoplasmic connection that is passing from one cell to another cell through cell wall. Within the cell wall there are gaps. Those gaps are called pits. Okay. The phloem parenchyma store food material and other substances like resins. That's what I say. Resins would be stored in phloem parenchyma. Tannins will be stored in xylem parenchyma. Okay. Latex and mucilage. Phloem parenchyma absent in most of the monocotyledonous. Very important. Phloem parenchyma need not to be present in monocots. Phloem fibers, which sometimes also called bast fibers, are made up of sclerenchymatous cells. It's not necessary to say fibers are made up of sclerenchymatous cells. Be careful. Wherever there is a presence of fiber, that fiber definitely should be present only for sclerenchymatous fibers. There is no another variety of fibers existing in the plant. Got it? These are generally absent in the phloem, but very important. They are generally absent in primary phloem. Which one? Fibers. and they are found in secondary phloem because secondary phloem needed more strength. These are much elongated, branched, very important, very important about phloem fibers. They are not found in primary phloem, but they are found in secondary phloem. What about phloem parenchyma? Phloem parenchyma absent in case of the monocots, but it is typically seen in case of dicots. That's the difference. These are much elongated, unbranched, have pointed needle-like apices. Apices means tips. The cell walls of the fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. That's of course fiber character is that. Phloem parenchyma of jute, very important. Jute, flax and hemp are used commercially. First named primary phloem consists of narrow sieve, that's called phloem, protophlloem, and if it is mature, that's called metaphlloem. Whatever the direction that you have seen, for example, I have seen there is a endark protoxylem. Endark protoxylem that you have seen in case of the stems. The same feature would be there for phloem also. Are you getting the idea? Phloem and xylem would be having same direction, which means xylem is endark xylem, phloem is exarch phloem. No. Such situation you will never come across. Xylem and phloem are work together. That's why they are called conducting tissues or conducting elements. Got it? Tissue system. Tissue system, the peripheral tissue systems, you can understand. We have three types of tissue systems. One is epidermal tissue system, ground tissue system, next one vascular tissue system. What about peripheral tissue system? We have epidermis. That's especially in the epidermis, we have some kind of epidermal modifications like stomata, root hairs and trichomes. Stomata, root hairs and trichomes are epidermal tissue modifications. Okay? Over there, they are talking about stomata. Do you remember some kind of structure we have seen there? Accumulate layer. Epidermis usually single layered. Underline that. It's not always single layered. It's usually single layered. Multi-layered epidermis would be also found in case of some hydrophytes, okay, which needed some more strength in the epidermis. Epidermal cells are parenchyma to small amount of cytoplasm. Very important. Small amount of cytoplasm lining inside the cell and a large vacuole. Outside of the epidermis often covered with waxy layer called cuticle. Remember, cuticle would never be found in case of root epidermis. It is typically found in young stem and young leaves. Next one, which is prevent the water loss. Cuticle is meant for the prevention of water loss and cuticle absent in the root. Very good. Stomata structures are present in the epidermis of the leaf. Stomata regulate the process of transpiration. What is transpiration? That is removal of water by gases. What I mean to say exactly, if we are removing water in the form of vapor, removal of water in the form of vapor, we will call it as transpiration. Along with the transpiration, stomata is also meant for exchange of gases. That means transportation of gases. Sometimes it would be removing carbon dioxide from the plant body. Sometimes it is taking oxygen inside, and variety of things will be happen. But typical tendency is this: a plant while performing photosynthesis, it takes carbon dioxide from outside to inside and it releases oxygen to outside. But when the plant is performing some kind of respiratory events, at the time if necessary oxygen is not there in the plant body, there would be an intaking of oxygen and even carbon dioxide will be released. That's why they did not mention particularly releasing of the carbon dioxide and intaking of the oxygen. Such kind of words they did not mention because the exchange of the gases would be varies depending upon the requirement of a plant. Next one. These two are called guard cells. This area is called stomata. Exactly, stoma means singular, stomata means plural. There are two guard cells. These two guard cells vary in shape significantly in between monocot and dicot. In dicot, there will be bean shape or kidney shape in dicots. In monocots, they will be dumbbell shape. That shape we can see, understand. But inner layers will be more thickened when compared to outer layers. They have some kind of special cells in their vicinity. Those special cells we call it as subsidiary cells. Subsidiary cells are a special thin parenchymatous cells. They will give water to guard cells in order to open and close the stomata. Exactly. Opening and closing of the stomata is due to guard cells. That guard cell is going to take some help from subsidiary cells. One more thing. Generally, epidermal cells do not have typically, okay, epidermal cells do not have any kind of chloroplast, but guard cells are the only epidermal cells which can have chloroplast. Important, that one. Chloroplast will be there in the guard cells. Next one. Cells of the epidermis bear hair. The hairs are unicellular extensions. The root hairs are nothing but unicellular extensions. These unicellular extensions will increase the surface area in order to absorb more amount of water and minerals. Next one. On the stem, epidermal hairs are called trichomes. Very important, trichomes. Remember, if trichomes are present, that stem is called hairy. If trichomes are absent, that stem is called glabrous. If somebody is asking a question to you, do all stem have trichomes? No. Answer should be no. Okay. But do all roots have root hairs? Of course, yes. If roots do not have root hair, the surface area would be reduced and they could not be able to get more quantity of water. Understand? But when it comes to stem, for example, there is a stem called bamboo. If you see the bamboo, they have very soft stem. The soft stem is called glabrous stem. If the hair would be seen on the top of the stem, that stem is called hairy stem. Got it? Next one. Ground tissue system. All tissues except epidermis. Trichomes are the shoot, very important, some amount of the things. Absorb water and mineral. Trichomes are also shoot, usually multicellular, maybe branched or unbranched, soft or stiff, may even secretory in nature. They could able to secrete resins, etc., etc. Trichomes help in preventing the water loss due to transpiration. Be careful. Root, for example, stomata. Stomata is meant for increasing the transpiration. If trichomes developed on the stem, that stem is not ready to be performing transpiration. Transpiration to be controlled with the help of trichomes. Transpiration would be supported with the help of stomata. Ground tissue system, except epidermis and other kind of remaining, all we will call it as ground tissue system. That would be common to us. Vascular tissue system. Observe here. Vascular tissue system. Xylem and phloem to be collectively called as vascular tissue system. This is about root, primary xylem, primary phloem. This arrangement of xylem and phloem on two different radius. For example, if you consider it is a circle, this is one radius. On one radius, xylem is present. Another radius, phloem is present. If xylem is on one radius, phloem is on another radius, that is called radial vascular bundles. Radial vascular bundle. Next one. If you take one vascular bundle, xylem and phloem are present on the same radius. This is called conjoint collateral vascular bundle. If conjoint collateral vascular bundles are having a cambium, this cambium you have to call it as intrafascicular cambium, or sometimes it is called fascicular vascular cambium. Fascicular vascular cambium. Such kind of vascular bundles are open. Open for what? They are open to conduct secondary growth. Later they can conduct secondary growth. These vascular bundles are called closed because they would not be able to perform any kind of secondary growth. There is another kind of thing is there, bicollateral vascular bundle. In the center there is a phloem or xylem, either one should be present, but I am taking here. In the center there is a presence of xylem, here phloem, here phloem. This is called bicollateral vascular bundle. Bicollateral. Like this, there is a development of vascular bundle systems. The radial vascular bundle is a characteristic feature in case of the root. Conjoint vascular bundles are characteristic feature to stem. But be careful. If anybody asking, what about leaves? Do leaves have vascular tissue system? Of course, yes. But the leaves will have the vascular tissue system that is similar to stem. Stem would have conjoined vascular bundle. The same kind of vascular bundles will be also present in case of leaves. Leaves do have conjoined vascular bundles. Next one. Anatomy of dicot root and monocot root. Here you remember the difference between the dicot and monocot root. I would like to explain. All are same, but the only difference is here you will have pith is small. This is pith is small because fewer vascular bundles are present. If 2, 3, maximum 4. Here pith you can see it's a larger one, somewhat larger. Okay, because vascular bundles are going to be more. Okay, more the vascular bundles, center area is going to be enlarged. This is the main difference. Here more than 4, here 2 to 4 vascular bundles would be present. Remaining all are same. Nothing to be worried about it. Next one. In case of dicot and monocot, you observe that in case of dicotyledonous stem, look at here in the dicotyledonous stem, what are the differences? What are the things we need to remember exactly? Epidermis, trichomes. Remember, monocot stems do not have trichomes. That's why I told you in the beginning there is a structure that is called as bamboo. Bamboo do not have, okay, secondary growth. Generally it does not show secondary growth. Next one. Trichomes are found, and after that collenchymatous hypodermis. In an young stem you will have collenchymatous hypodermis. After that general cortex. Okay, after that endodermis. Endodermis of dicot stem we call it as starch sheath. Starch sheath. Why did you call it as starch sheath? It could be able to store some grains of the starch. That's why it is called starch sheath. Next one. After that pericycle. Pericycle, this is very interesting. Pericycle is semilunar. Semilunar, or we can say crescent shape. Crescent shape. Crescent shape pericycle. The pericycle is the one which is also called bundle cap, because it will be present on vascular bundle. Okay. After that phloem, xylem. Between xylem and phloem, there is a presence of intravascular chemium, as that we have noticed earlier. Protoxylem towards the center, metaxylem towards the periphery. This condition is called endark-xylem. Pith is large. You can see here the large amount of pith. Next one. All the vascular bundles are arranged like a ring. The ring-like vascular bundle is called eustele. Eustele. The arrangement of ring, that's called eustele. Ring-like arrangement. Next one. What about monocot? This is should be remember in case of monocot and dicot stem. You remember this. Epidermis. Hypodermis is not parenchymatous. Generally sclerenchymatous or collenchymatous. Collenchymatous hypodermis is a characteristic feature of dicot stem. Next one. Below that there is a ground tissue. In the ground tissue, vascular bundles are going to be scattered. Very important. Vascular bundles are scattered. The scattered vascular bundle, remember this. One time I will explain why they are scattered and why that necessity is to be there. Observe, all the vascular bundles would be scattered because in case of monocot stem, there is no secondary growth, and also there is need of more kind of vascular bundles when compared to dicot stem, because they have soft nature inside and most of the area covered by living tissue, that's called living parenchymatous tissue. If more the parenchymatous tissue inside, more the amount of food and water should be supplied. In order to do that, they have peripheral vascular bundles are smaller, which are scattered anyhow all over the area. Central vascular bundles are larger. Central vascular bundles are larger because they are matured. Matured. What do you mean by matured? They have a cavity that gradually enlarge. That's called water-filled cavities would be found. That a water-filled cavity gradually enlarges and make the vascular bundle to become enlarged. When the vascular bundles enlarge, automatically they can conduct more amount of water and food supply. That's the reason why they have large sized vascular bundles in center, which are able to conduct water. Matured ones they are. Next one, peripheral vascular bundles are smaller. The smaller peripheral vascular bundle generally meant for mechanical strength. They are meant for mechanical strength. Got it? Next one more feature is there. They have bundle sheath. The bundle sheath of monocot stem is also sclerenchymatous. Remember. Okay? Next one, monocot leaf and dicot leaf. Observe this. Upper surface is adaxial surface, lower surface is abaxial surface. This surface towards the stem, if it is a leaf, okay, this side is adaxial surface, this side is abaxial surface. Remember, towards the top xylem is present, towards the top xylem is present, towards the bottom phloem is present. Phloem is present towards the abaxial surface, the xylem is present towards adaxial surface, because leaf has been separated from the stem. In the stem, imagine this is a stem right now. Gradually a piece of stem would be separated. When a piece of stem has separated, whatever in the center would be coming to the top. Whatever it present peripherally would be coming to the down. Getting the idea? That's why there would be a presence of the such kind of thing. Remaining things would be already there. Okay, one thing I would like to say, secondary growth, if you are interested, go through it. If you are not interested, that's your choice. You have seen the paper. There they would not stress about secondary growth, because secondary growth is not there in the new NCERT book. If they give any questions from there, it will generate lot of conflict. Okay, any more doubts? Come on. Somebody is asking, online better or offline? That's your choice. Okay, if you are comfortable with the screen and the relevant things associated with the online program, that's your choice. If you are not comfortable with the screen, and also if you have a habit of constantly skipping away from the regular thing and watching the unnecessary thing, it's better to go for offline. Okay, yes, in the next class onwards it would be operated on app. Protoxylem, remember, if I take, somebody is saying protoxylem location, this is stem, right? Okay, I would separate it and I will remove. So upper side would be, observe this location, upper side, this is center is protoxylem, right ma? Peripheral is metaxylem. So adaxial surface protoxylem, abaxial surface metaxylem. Somebody written here, in palms, monocotyledonous plant, as they grow very tall and increase in the girth in massive trees, since monocots lack lateral cambium, how could they achieve this thickness? The thickness would be achieved due to, okay, abnormal secondary growth. That's called anomalous growth. There would be some amount of intercalary meristem, right? That intercalary meristem would regain the strength to give more kind of fibers. The fibers are the one who make the plant to be enlarged. Especially if you talk about coconut also, coconut is a monocot. Palm tree is also coconut. Palm tree is also monocot. These palms and coconuts and bamboo, they will get more fibers. More the fibers, more the amount of mechanical strength. In simple and complex tissues, is there any syllabus? Yes, it is there in the syllabus. You have to study. Okay. Somebody saying here, intercellular spaces and pits are different. No. Pits and intercellular spaces are totally different. Be careful. Pit is a gap in the cell wall. cell wall of one cell has a minute pore. That pore is called pit. Intercellular spaces, the difference between one particular cell to another particular cell. Okay, they are saying assignments, you would be getting assignments and the regular test in the name of dat in the coming next day onwards. Next one, turgid means filling of more water. If you, for example, you are filling a balloon with water, while we are filling the water inside the balloon getting enlarged, that enlargement of the balloon we call it as turgidity, water strength. Sir, dicot root will undergo secondary growth. Of course, dicot root will also undergo secondary growth. Imagine the stem is getting enlarged. Enlargement is there in the stem, but root is not enlarged. Of course, the plant is going to be fall down. That's why stem and root simultaneously undergo secondary growth. Collateral means one. For example, somebody says, what is collateral? This is a vascular bundle. Okay, this is stem. For example, one vascular bundle, another, one more. Let me take here simple. They have given it, right? Observe. This is a dicot stem, right? Okay, in the dicot stem, this is a vascular bundle. This vascular bundle, if I draw it's a circle, right? On that circle, I am going to draw a radius. On the radius, xylem is in the one side, phloem is on another side. This is called collateral. Next, what is bicollateral? Let us take the same kind of vascular bundle. Okay, here xylem or phloem. I am taking here phloem. One side xylem, another side xylem, or else one side phloem, another side phloem. Either direction you can take. This we will call it as bicollateral. Only one patch, collateral. Two patches are there alternatively, bicollateral. Okay. Somebody said here something. Secondary growth should be study or not? You just go through it once, but don't give that much importance. You just go through it once. No need to dig into the details. Make sure that you don't find any question. Completely debark the commercial woody tree. We remove the, somebody say, could you send that bark some kind of thing you are saying? Could you resend it? It has gone up. Schedule will be there. Okay, you will get the schedule. Somebody said bark or something. If we completely debark a commercial woody tree, we remove the peridome and secondary phloem. If the water conducting xylem is completely untouched, why does the tree still die after few days? This is called as griddling. Imagine if I take a well growing dicotyledonous stem and I am removing its bark. When bark is removed, okay, I am removing the bark, entire circumference I am removing. If they removing the bark, the food transportation will be hampered from the top to bottom. So if the food is not there for the lower parts, lower part will be weakened and gradually die. That ultimately upper part also will going to die. This phenomena is called griddling. That's very good. Cork cambium and bark cambium are the same. There is no word, I think so, bark cambium. Okay, you consider it's a bark cambium is also not official word. Cork cambium is official. Bark is not official term. Got it? Bundle sheath cells present in all except dicots. Bundle sheath cells. Bundle sheath cells. No. It's not like that. Bundle sheath cells could be even seen in case of dicot and monocot, both. Bundle sheath means those cells, those parenchyma, sclerenchyma, whatever it may be, which are covering around the bundle sheath. I mean vascular bundle. That's called bundle sheath cell. They are not specialized bodies. Okay. Yes, radial will be generally seen in case of root. Okay, our time is up. If anything is needed, we will meet in the next class. Is there anything you needed? Exactly. Inconspicuous. Inconspicuous means not seen clearly. Inconspicuous means not seen clearly. Is it okay? Okay, done. Thank you. We will meet in the next class.